Namaskar दोस्तों, welcome to channel Toxicology on Board. Today we'll discuss question number 38, and for this video the topic will be DABT question, dermal absorption and animal models which are used for dermal absorption. So let's directly dive into the question. This question, as usual, this question is taken from American Board of Toxicology, Design, Execute and Interpret Toxicology Study Quiz. What animal model is considered the best predictor of dermal absorption of chemical for humans? Okay, the question is asking we have to choose the animal model which is the best predictor of dermal absorption of chemicals. Okay, in for humans. Option A is rat. Option B is pig. Option C is mouse, and option D is rabbit. So as we all know that all these things, all these rat, pig, mouse, rat, rabbit, they are the animal model to predict certain toxicological endpoints. So out of which we have to identify which is the best model to do dermal absorption. Okay, as you can see, the answer will be in green. Okay, the right answer is option B, and the explanation and reference which is given by American Board of Toxicologists is historical research has shown that in general chemical preparation penetration of human skin is similar to that of pig or monkey. Okay, it is saying that human skin penetration is similar to that of pig and monkey and much slower than that of rat and rabbit. So, in case of rat and rabbit, dermal penetration is high, but human skin, as well as pig and monkey, the dermal penetration is less. Okay, so henceforth, before going in detail about the question and explanation, we'll also confirm whether this question is still there in the current DABT syllabus or not. Okay, to do that, what we have to do is first we have to go through DABT brochure, that is year 2025. Okay, those people who are appearing, those toxicologists who are appearing for the exam for 2025, no, definitely they might have gone through the brochure. Okay, so in that you please check whether this question is still there in the syllabus or not. So I have tried to, because exactly you will not get whether this question will be, because the syllabus is as such is very expansive, around 3 to 4 pages will be there on the syllabus only, topics only. Okay, then references to the books as well as guidelines will be another 2 pages, something like that. So I thought that probably this question might fall into this particular page number 27 of 34. You can check effect of species differences uh, in anatomy and physiology on uptake and absorption, respiratory tract, dermal and oral. Probably this question might fit into this particular section. Okay, so the so generally, as what we do when we go through a certain question, we will try to learn all the concepts related to this particular question. Okay, with the help of Clayson or other toxicology as well as guidelines. So first let us understand the cross section of human skin. Okay, for this please go to Clayson 8th edition, chapter 5, page number 166 and as well as 167. Okay, in that you will see the different layers in the human skin, like and the cellular composition and blood supply. Important thing that you have to remember is stratum corneum, which is the outermost layer of the skin, and stratum germinatum, which consists of stratum spinosum and stratum basilae. Okay, epidermis, dermis and subcutis hypodermis, you try to remember. Also you try to remember sweat gland. Okay, and erector pili will be there, that you try to remember, and sebaceous glands, hair follicle. So these are the these are the components, various layers in the skin that you have to remember. Other components layers please read in detail. You can either refer to Clayson, or those people who are pharmacy students, you might have already learned about the skin histology or skin various layers in your anatomy and physiology books like Totor and other books. You can go back and go back to your basics to understand the various components of the skin. Okay, now let us directly go try to remember what are the important points in the case of absorption of the skin. The extent of absorption through the skin is highly dependent on the lipophilicity of the compound. As we know that if a skin layer is made up of cell layer is made up of phospholipid bilayer, no, based on lipophilicity, hydrophilicity of the molecule or a chemical, the cell membrane penetration takes place. Similarly, in the case of skin also, the extent of absorption it highly dependent on the lipophilicity of the compound. Okay, stratum cornea, uppermost epidermis layer. This is the stratum corneum. Okay, provides an effective barrier, right? Stratum corneum cells are mostly keratin. So try to learn what is the composition of the keratin, outermost layer which protects the main function of keratin is to protect. Okay, absorption through skin is mainly by passive diffusion. As we know, from do you have different cell transport mechanisms are there, passive transport, active transport is there, facilitated diffusion, active transport where energy dependent or ATP dependent will be there, whereas in the case of passive diffusion it is concentration dependent. The movement of the chemicals or ions takes place across high concentration to low concentration. So similarly absorption through skin is mainly by passive diffusion. The skin does not appear to have active transport mechanisms. As I said, no, there is no ATP mediated or active transport mediated mechanism is not available on the skin. Okay. Next, the rate limiting step is crossing the stratum corneum. So this is the stratum corneum. Cross any chemical crossing this through layer. That is the main rate limiting step. Okay. This is the one which is going to decide whether a substance can easily penetrate or slowly penetrate. So that is it is called rate limiting step. Stratum corneum is very thin in the scrotum and compounds can cross readily. So again you have to remember one thing, throughout your body, skin is not distributed equally. In certain regions it is very thin, in certain regions it is very thick. So in case of scrotum region, stratum corneum is very thin, so any penetration of any chemical or compound through this particular layer is much easily, compounds can cross readily, right? stratum corneum uh, stratum corneum is very thick on the soles of feet and palm of the hand. So here you can see hand and here you can soles are, here the stratum corneum layer is thick, hence the penetration is very difficult. Compounds do not cross as readily in these areas. Okay, this point you have to remember. Next, the stratum corneum is less permeable when the water content of stratum corneum is reduced. So, the permeability of this particular layer also depends on the amount of water content, okay, in this particular layer. Okay, if the water content in the stratum corneum is less, the permeability is less. So, the water content in the stratum corneum is directly proportional to permeability. Like this you have to remember. Okay, can increase the permeability several fold. Hydrating the stratum corneum, if you are able to increase the water content in this particular layer, then you can also increase the permeability of the by several fold. Okay, next, guinea pig. This comes to our question. Guinea pig as well as monkey skin is about as permeable as human skin to many compounds. So this is very important point. Whereas rabbit and rat skin are often more permeable than human skin. Okay, if you see the, if you make a comparison this thing, no, you can put rabbit is greater than rat or like that, then afterwards this will come your either pig or guinea pig or human, like this, greater than like that. Okay, then coming to important thing, in a historical incident that we have to remember with respect to dermal absorption, which we have seen in the first slide about the scrotal thing, scrotal the thin this stratum corneum in the scrotum region is thin, no, based on that one historical incident is there, that you will also try to remember. So that you can see in Claysen's ninth edition, chapter 1, the evolving journey of toxicology, historical glimpse, page number 9, you can go and check what exactly happened in this incident. Is that in the 18th century, British surgeon, okay, Sir Percival Spot first linked scotter cancer, also known as chimney sweep cancer, to occupational exposure to soot in chimney sweeps, establishing the first document link between occupational exposure and cancer. So this particular incident is also important with respect to occupational toxicology. Why? Because this was the first incident where they have made a link between the incident as well the cause for that particular cancer. So this particular scientist Percival Pot, he was able by giving lot of publications, other papers, and uh, he was able to establish this link. Okay, you can go back and read in detail. It was very unfortunate for these kids because they were very young kids. Okay, and if you see in case of British or London, no, the the chimney sweep is very long and thin. Children were able to penetrate down and clean the chimney sweeps. Okay, and most of the time these sweeps, these chimneys, they contain definitely black soot because the combustion of that wood or other materials. Okay, and this black soot had many components which caused cancer, scrotal cancer or lung cancer, other type of cancer, skin cancer. This type of cancer it used to cause. But another important thing that you have to remember is that it was until 1920 that benzo alpha pyrin was identified the actual chemical responsible. Initially they thought that soot is the one which is responsible, black soot is causing that cancer. But now with, you have to understand, science is always keeps on evolving, more and more literature comes. Okay, initially they were not able. See, black soot contains of many constituents, many components will be there, out of which one of the component is benzo alpha pyrin. We have para PAH also will be there, which also causes various types of cancer, which is also important component in the case of black soot. But benzo alpha pyrin is the one which is responsible for causing this cancer. Eventually they have able to explain it in 1920, was identified the actual chemical responsible. Okay, you can go back and read this in detail. Okay, now let's directly move to another slide. Absorption, because we have to not also deviate from the topic. Topic was absorption of toxicant through the skin. Okay, skin is not permeable and is relatively good barrier. Okay, we have various things here. But you have to remember, blood brain barrier is the most difficult barrier. But another thing what they are saying is skin is also good barrier. uh, uh, Relatively it's a good barrier, not permeable to all the things. However, certain toxicant can be absorbed in sufficient quantity to cause toxicity. To be absorbed, chemicals must pass through epidermis and appendages. So that is the reason I have discussed in detail about the various sections of the skin. So epidermis, appendages, I was saying, no, sweat glands, sebaceous glands, and hair follicles. Also, you have to remember because they are secondary barriers. First one is stratum cornea, which is a rate limiting step. After the second barriers will come these points. Epidermis consists of stratum corneum, stratum granulosum, stratum spinosum, and stratum germinatum. Okay, stratum cornea, horny layer, uppermost layer of the epidermis. Okay, rate limiting barrier, first phase in the case of skin or dermal absorption. Okay, replenish this particular stratum cornea, no, every 3 to 4 weeks in adults it keeps on replenishing. New layer of skin will be formed. New layer of stratum cornea will be formed. Okay, next involves gross dehydration and polymerization of the intracellular matrix, keratin, filled dried skin layers. So that's why keratin is the most protective layer, you can say. Okay, our hair follicle, our nails, they are made up of keratin only. Next, partially hydrated, normally 7% by weight, but can less when dry and can increase weight by 3 to 5 fold when comes in contact with water. Okay, next, permeability increases with increasing hydration. In the previous slide also we have seen. Okay. Next, toxicants must diffuse through the stratum corneum because this is the rate limiting step. Next, we'll see what is the nature of the particular chemical. Polar and non-polar components are going to affect the permeability. Polar compounds diffuse through outer surface of the protein filaments. First you have to understand what is the what do you say, what are polar components and non-polar components. See, polar components depends on the charge of the particles. They are unequally distributed. Okay, they have negative and positive ends will be there, whereas non-polar components may negative and positive charge are equally distributed. So this nature of that particular chemical is going to decide whether a substance can diffuse at a faster rate or slower rate. Okay, polar compounds diffuse through outer surface of protein filaments. Non-polar compounds diffuse through lipid matrix, phospholipid bilayer between the protein filaments. Okay, rate is the proportional to lipid solubility and inversely proportional to molecular weight. As the molecular weight increases, the diffusion through this particular layer will be decreased generally, but also affected by type of lipids. Again, see, for any statement, no, there will be some exception will be there. Okay, it is saying it depends on the phospholipid bilayer. No, it is saying based on lipid solubility. Again, you have to see that that lipid layer also made up of different types of, like, triglycerides, cholesterol, ceramides, etc., which varies between each and every species, right? Mice, rabbit, dog. Okay, Next, this stratum corneum only we are still discussing, composed of 75 to 80% moderately lipophilic materials, varies in structure and chemistry from one region of the body to another. Example was called, which affect the permeability. Permeability depends on the diffusivity, that is passive diffusion, which we discussed, and thickness of stratum corneum. Lower levels of epidermis, stratum granulosum, stratum spinosum, stratum germinatum, and dermis, sweat ducts, sebaceous glands, blood vessels, connective tissue, fat, hair follicle, and capillaries. These are the secondary layers actually. Second phase of absorption must pass through these cycles, through these layers. Not a good diffusion barrier, porous, non-selective, aqueous. Unlike stratum corneum, these are porous, non-selective, and aqueous. And rate of diffusion depends on the blood flow, interstitial fluid movements, and interaction with the tissues. Okay. So these are some of the important factors that you have to remember. It depends on, so rate of absorption, dermal absorption basically depends on integrity of the skin, whether the skin is intact or abraded. Certain times what happens, if your skin layer is damaged on the uppermost layer, if it is damaged, the chemicals, if you get exposed to that damaged skin, abraded skin, more penetration will be there, unlike unabraded skin or intact skin. Okay, so that is one of the important factor. Conditions of the skin, as we said, hydrated and dry skin. Hydrated is directly proportional to permeability of that particular component. Then location of the skin. Okay, then type of toxicant, polar or lipophilic. So these points also I have already discussed. Okay, species difference affected by lipid content of the skin, thickness, number and types of appendages, and blood flow. So why I am trying to explain so much again and repetitively is that how pig is more favorable when compared to rat or rabbit or dog. Understood? Most selective species to understand dermal absorption, because the structure of the skin in the case of pig, guinea pig, or monkey is similar to that of, so these are the components which might be factors which may be similar in the case of human. Okay, then along with this topic, we'll also, I wanted to bring in this video so that you people can make a note and read at your end, that your examiner, ABT examiner, might ask you some questions with respect to alternative models which can be used to do dermal absorption or dermal toxicity, you can say. So I have listed out this OECD 427, OECD 428. So 427 is skin absorption in vivo method and skin absorption in vitro method. See, 427, 428 are not directly given in the case of brochure. Okay, they are not directly given in the case of brochure, but they say that OECD test guidelines, few of the guidelines OECD numbers are mentioned, but not these two were mentioned. But I am not sure if they are still, they might ask or not. Then also I have given you guidance document and dermal absorption. Okay, you can see OECD guidance document 156 also. And I also given this one, which is from NTP site, ICCVAM, which is all alternative methods which are used, because nowadays animal testing is not recommended. There are a lot of alternative methods are available, to do those studies. So ICCVM with respect to those alternative methods also, one of the interesting topic or favorable topic for the questioners to ask. So if possible, you can go back and read these particular guidelines or test, try to understand. So these alternate methods, how can be they useful for a dermal absorption prediction, or to give a absorption percentage, you can say. Then next we will also see, so finally we understood that laboratory animals are typically not good models for human absorption of toxicants. Species difference and lack or concordance with human data for dermal absorption. So this is again very important thing. Clayson 8th edition, chapter 5, page number 166, you can see how they are giving an example with a case study and saying that which of the following models are more favorable for the dermal absorption. If you see, they have taken an example 2,4-D, which is an herbicide. Okay, it was a part of Agent Orange. Those who people historically, again it has certain significance. Those this was Agent Orange which was used in Vietnam War. In that, one of the component I'm saying, but the one which caused teratogenicity or toxicity, that is again different compound. TCDD it is there, which is a contaminant in the case of Agent Orange. But 2,4-D was a important one of the component in the case of Agent Orange. So like that they will take examples, but let's not again deviate from the topic. Vietnam War and the TCDD, that about teratogenicity, etc., we will read in another video, or another topic will take by itself. But sticking to this particular table, 2,4-D is the herbicide. Okay, and they are seeing the dermal absorption across various species: rat, rabbit, monkey, and humans. Okay, this is a wash time. Rat, rabbit, and monkey, they applied on this animal skin. After 24 hours exhaustion, they washed it, they removed it. Okay, and they have collected the samples at so and so time points they have given. But in case of human, the exposure time was only 4 hours. They have removed it after 4 hours, and collection time was 120. And if you see percentage of absorption, rat is having highest absorption, 20%, next rabbit, then monkey, then human being. So this part you read how much area of exposure will be there, because when compared to oral and other drugs, it will be, the units will be in mg per kg, but in case of dermal products, it depends on the body surface area. Similarly, in the case of anti-cancer drugs also, it also it depends on bsa rather than body weight. So area of application, all these things you read. But final conclusion is that human being absorption is 3.7, which is similar, somewhat closer to monkey. That is what the point is. So you read the sentence in detail. So okay, higher than other species, primate or pig skin is considered most closely predicted human dermal absorption. Okay, additionally, this is another final point that you have to keep it in your mind is, in vitro methods using human skin may provide reasonable alternatives to use of laboratory animals. As I said, no, you should go for you should look for alternative methods. Okay, so in that they are saying that human skin is very good alternative model for evaluating dermal absorption of toxicant. So for that, human cadaver skin, also known as allograft or homograft, is donated skin from deceased individuals. Okay, used for medical purposes such as temporary wound coverage or studying percutaneous absorption. So hope you have learned from this video about dermal absorption. What are the animal models, what are the factors which are considered or kept in mind, rate determining steps, importance of stratum corneum. Okay, and alternative methods, what are the OECD guidelines for dermal absorption. Okay, so uh, thank you so much. Be curious. For any questions, please comment. You can always write a mail to arya.priyanka18@gmail.com. Like, subscribe, and follow my YouTube channel, Toxicology on Board. All the best for your exam. Thank you.